If you've been listening to the Business of Biotech podcast for a while now, you'll recall that Erin Harris has joined me to co-host a few episodes. Erin's my friend, colleague, and chief editor over at cellandgene.com, and she just recently launched a podcast of her own. It's aptly named Cell and Gene, the podcast. And if you're working in the cell and gene space, you should give it a listen. It's a collection of interviews with the industry and academic leaders moving the space forward. And you can find it at cellandgene.com or anywhere you listen to podcasts. Cell and Gene, the podcast. Check it out. So as we sit today, just 4 years after the first gene therapy was approved by the FDA here in the US, the gene therapy manufacturing story remains largely unwritten. It's an area of constantly new discovery and innovation, and that paints a beautiful landscape for discoverers and innovators like Dr. Palani Palaniappan, Chief Technology Officer at Aruvant. On today's episode of the Business of Biotech, we're taking a deep dive into the trends shaping the future of gene therapy manufacturing with Palani, a man who's led development and manufacturing efforts at Gilead, Takeda, Sarepta, and now Aruvant. Joining Dr. Palani and I for the conversation is Project Farma EVP Tony Khoury, a longtime friend and contributor to Bioprocess Online and a biopharma manufacturing Sherpa in his own right. Tony's contributed to manufacturing operations and leadership roles at Boehringer Ingelheim, Amgen, Sandoz, Gilead, Avexis, and more. Dr. Palani and Tony, welcome to the show.
Thanks for having us. It's great to see you again, Matt.
Oh, it's great to have you guys. I appreciate the time you're taking to spend with us. So let's rock and roll here. We have a lot of ground to cover. We chatted before the recording. We've got a lot that we want to cover. So, Palani, I want to jump right in with you and ask why manufacturing, more specifically manufacturing systems and processes, is so critical in gene therapy and where we are at this juncture.
Yeah, absolutely, Matt. It's a great question. Before I get there, let me just very briefly introduce, myself and my company. We are a gene therapy manufacturer for rare disorders, and we focus on sickle cell disease to begin with, and expanding the pipeline into other rare disorders. And it's great to be here today, and especially, with Tony speaking and reminiscing with where gene therapy is going. Now, to address your question, why is this important? This time in gene therapy reminds me of late 80s and 90s, the inflection point in biologics therapeutics, antibodies, proteins, a range of biologics options came into play for patients in those time frame. And this feels like one of those inflection points in the industry where, we get to address multiple types of diseases, rare disorders, genetic disorders, in a range of diseases. I think that the important piece that we all have to acknowledge here is the curative potential of gene therapy. When we talk about biologics, other therapeutics, it's only intervention. But at this point in time, we have, I think, enough evidence to say that gene therapies and cell therapies give you an opportunity to cure a disease. And that's the excitement. Therein lies the reason why we feel the patients will benefit from it. And a lot of these options, we had questions about safety, but that seemed to be going away as well. These therapeutics that are coming out are generally appear to be safe and efficacious, and all it takes is one dose. Who would not like to have just one dose and cure a disease? And therein lies the reason why there is so much excitement. Just to give you a little bit of statistics, there's about 1,100 or so companies now thinking about gene therapy. Billions of dollars are flowing into this space from venture capitalists, others who want to invest in this space. There's manufacturing activity happening all around. Companies are putting a ton of money in enhancing manufacturing capability, capacity. Tony can talk to this a little bit more than I. I think there's so much excitement simply because this is curative, it's safe, and there's all sorts of opportunities to treat and cure a range of diseases using these approaches. Tony, anything you want to add there?
Yeah, thanks. Palani, and it's great to see you. And I mean, where to begin, right? It's a very critical time right now, and you hit the nail on the head, right? We're humbly, all of us are privileged to be a part of the advanced medicine space, the therapeutic revolution. And with that, there's so much beautiful science out there that we need to mobilize. And in order to do that, we have to continue to evolve and further on with, how we progress the science and the technology. I mean, looking at a few of these, diseases that have now been cured and being, humbly a part of it, seeing the Zolgensmas, the Yescartas, the, Kymriahs and Luxturnas. I mean, they're beautiful stories. And we're not just saving lives, we're creating lives, because now those patients can have families or further on their families. And it's truly, humbling to be a part of it. From a manufacturing look, we've had tons of clinical trials. It's continuing to grow year over year. We've seen the investment, we've seen the science, preclinical, clinical continue to grow. Well, what we keep forgetting about is now everything's going to progress into commercial. And we've had a handful of commercial products move forward, with many more in the pipeline, right? As we continue to see clinical success. And so companies, continue to plan for success. We need to plan for success as an organization. And we really need to be able to address the manufacturing options out there and find solutions that would meet, and do justice to the great science that we have and the therapeutic revolutions that we see.
Yeah, I'm curious to get both of your perspectives on something that you mentioned, Dr. Palani, around, sort of the parallel between biologics back in the 80s and now where we are, the inflection point that you see happening in gene therapy. If you kind of took those two chronologies and laid them over one another, like this inflection point in biologics back in the 80s and where we are today, that kind of correlates with that in gene therapy. The trajectory of the manufacturing challenge, or the curve, I should say, of the manufacturing challenge. Do you see a steeper curve for gene therapy where we are right now than where we were at sort of a parallel time in biologics, because of the complexity and the I guess lot to lot consistency and some of the major challenges that we faced? Do you see a steeper curve right now than we had back then?
It's a wonderful question. I think you touched on this point very well, in that the expectations of people who have diseases changed, right? People expect immediate cure. We've seen cancer explode in an exponential fashion in the population. So people want answers. So because of that, I think the curve is in fact steeper than what we saw in the 80s and 90s. Because remember, in the 80s and 90s, we had time. We had technologies that we had to build. We had to build manufacturing plants. We had to build these 25,000 liter tanks where we produce these proteins. We don't have that time anymore, because these diseases are, cropping up in a very quick fashion, and people expect solutions. And clearly the curve is steeper. And I think we are also responding to those expectations better these days, because our technology base has evolved as well from the 80s and 90s. So I think we have better tools, we have better solutions, and we have great quality people coming into this space as well. I think that sort of makes a difference in sort of saying, yes, the curve is steeper, but I think we can react to that steepness even better than what we did in the 80s and 90s. That's my response. I don't know, what do you think about that, Tony? But that's
no, I think it was well put, Palani. I mean, it's there, it's deep. The urgency is key, right? I mean, this is patients are passing away. There's no other options. There's no treatments. We 10 plus years ago, 15 years ago, we only dreamed. We had devastating stories, people dying of cancer, people dying of genetic diseases. Make them comfortable and enjoy the time you have. And now we have hope, we have cures, we have miracle medicines, miracle stories. So the urgency is there, and we can't just say there's no technology. We need to find solutions. And I mean, working with, connected with Palani 5+ years ago, and working with some of the first ever cell and gene therapies to ever be commercially approved, our motto was egos at the door, be solution driven. Mm-hmm. So find it out, make it quality compliant, make sure that we're safe. The profiles are there, but let's find solutions. Let's do justice to the families and the patients that are out there that need this help. I mean, those are the words that founded Project Farma when we started rock and rolling. The words are patient focused. That's where the PF for Project Farma came from. And we were like, hey, if we're going to embark on this journey, we want to do right, and we want to find solutions and move this medicine forward. Because if that was any one of us, I would hope that people would work hard and care and want to deliver.
All right, so I want to get your perspectives on scale up, and kind of get into the meat of the manufacturing discussion. I've had, as you guys know, I spend a lot of time talking with the leaders of emerging biopharmas and more specifically, many leaders of emerging cell and gene therapies. And, there seems to be, not a whole lot of middle ground between the folks who say, look, there's no way that we can outsource what we're trying to accomplish here in the development of our therapeutic because it's that complicated, and we want that in-house mastery. And then there's the other side of the spectrum who's like, you have to be crazy to try to do this stuff in-house in today's day and age. There's so much CDMO capacity and knowledge out there to leverage, and there's the whole cost consideration. So it's like a big dichotomy. It always surprises me, folks who are just convicted on one side of that or the other. Tony, what do you think? I mean, what's your take on, whether therapeutic developers will, are better off to scale up internally or to leverage CDMOs or some sort of combination? What does the future look like there?
Yeah, I appreciate the question, Matt. And it's a question that will continue to be a critical one for innovators. I don't think it's going to all day, every day. I feel like that's all I do is evaluate that with, innovators and lead our strategy group that does that. And I like to break it out into like two buckets. And one is a quantitative assessment and the other one's a qualitative assessment. Quantitatively, there's the financial benefits and upfront capital and cash flows and commitment plans and what's your return on investment and how many years for your break-even point. Sorry to go fast, but you can see me do it all day. But qualitatively, it's like, okay, control over your manufacturing, control over your fate. What do those lines really mean? And sometimes we just say those general lines, but we don't really understand what they mean. And the fact of the matter is, and Palani knows this, and so do you, Matt, like there's so many unknowns when we're first trying to move a product forward. We're preclinical, we have 20 some products or early phase clinicals. We don't know the yields. We don't know what scale we're going to produce at. We don't know the dosing volumes yet. And all of these are key items into determining how much manufacturing, how much GMP material we need to progress through clinicals and then position ourselves properly for pivotal and commercial manufacturing, right? So we have to put assumptions in. And those assumptions, I'll tell you just to assure everybody, they won't be right? They're going to move around. It's okay. But we need to put something in and then take that forward from there. And so based on those inputs and those critical assumptions, you make a strategy and a plan. More times than not, it's very rare that I work with an innovator or pharma company that says, I have 2 years. I can get manufacturing in a couple of years. It's almost always, and Palani knows this, it's like, I need something ASAP. So more times than not, to build a facility day one is going to be challenging. You need to partner with a good company, a good CDMO, but you also need to make a full-on strategy. And for someone like me, like living and breathing the tech ops world for my whole career, it's very interesting because the last 5-6 years being pulled in as early as private equity investment level. We haven't formulated the company. That's how important this decision is. They want a manufacturing strategy before they even create the company or invest in a company, or when they've invested and only have a handful of folks like the Avexis story that humbly be a part of. We were figuring out that plan so early in its infancy of the organization. So with that in mind, that shows you how early we need to plan. So it's kind of like you need to put out the fire in day one, and then you need to make the overall plan so you're preventative for the whole company with inputs that are moving around. So it would behoove you to evaluate that quantitatively and qualitatively, because when things do move around, can you adjust? Or is it hard to do that with a partner? Could you turn on those burners or run another lot if you need to, or bring on more material, or adjust the process so that you can get proper scale, scaling and yields. And there's a lot of things to take into consideration, and those are inputs. And then from a manufacturing side, people always just think manufacturing, but I want to just highlight to the team, there's analytical development, there's process development, there's manufacturing, quality controls, warehousing, supply chain and logistics, right? and critical long leads across the entire breadth. So when we say the word manufacturing, we're genericizing all the subcategories of analytical development, process development, warehousing, cold storage, manufacturing, quality control testing. That all needs to be assessed across both those two groups.
It's a good consideration. I appreciate you bringing that up, Tony, because I mean, it's easy to overlook that. It's easy to just say, oh, CDMO, just manufacturing outsourcer. We're not,
Right. So we'll make our product. And I mean, Palani's had tons of experience, managing the largest external solution in the space in gene therapy, and, as well as leading and building out capability internally. And, we always keep ourselves disciplined, right? It's not just manufacturing. We got to think about all these other buckets, right? Because it's critical.
Well, so I think this also goes back to the previous question Matt was asking about the steepness of the curve, right? I think the expectations are through the roof, and people need solutions. And, remember, building manufacturing plant and bringing it online takes 24 to 36 to 48 months, depending on who's doing it, where you are doing it, what regulatory considerations you need to bring in-house and things like that. So you don't have oftentimes that amount of time. So the question is, what's the best solution? Obviously the best solution is to have your destiny in your hand, build your facility, put in the money and all of that. But oftentimes you tend to settle down on the next best solution is to go to the external vendor and you pick the best one that you can find and get the solution going for the patients. And that's what happens oftentimes in these smaller companies. And remember, many of us are limited by the resources, right? We are coming into this with a focus on innovation, and you can only do so much for $100 million, right? If I had the big pharma approach and the muscle behind me, I can do a lot of different things. Even big pharmas these days, they're not taking that approach. But in a small environment with a steeper curve and the expectation through the roof, you often look for those hybrid solutions that Tony was talking about earlier. I think that's where many of us are at this point in time.
Yeah. So,
and just real quick, Matt, to pop in there is hybrid solution will happen. There's going to be more phased appropriate builds. So we've been seeing that, and we've been recommending that a lot because of these moving inputs, right? So it's, maybe you take a 150,000 or 200,000 square foot facility, but you only build out in phases. And then also with the CDMO, you partner in phases. So I think phase strategy will definitely continue. But it's going to be a critical decision. And de-risking that with a hybrid solution will be valuable.
Palani, can you give us sort of a snapshot of where Aruvant is in that journey?
Yeah, absolutely. Thanks for the question. I just to back up a little bit, I think we are a small company, and we are part of Aruvant family of companies. And so the resources are in some ways unlimited for us because we have the backing from Aruvant. But that said, we are focused on two disease paradigms today. So we are focused on sickle cell disease. As you know, sickle has been a much ignored disease paradigm for a long period of time in our society. More recently, in the last 10 or so years, number of companies are working on it, and we are in the mix of that. And we believe that we have a differentiated approach to be able to tackle that issue for the population. So our program is advancing to pivotal stage next year. We are in phase 1-2 trial. We have a few patients worth of data. Our program is in a place where we think, we like our chances. Let me put it that way. We have a hemoglobin, anti-sickling hemoglobin, which is much differentiated from what others have. And we have a conditioning regimen, which is very, very low intensity conditioning regimen, unlike the others who use busulfan, which is a painful way to deal with conditioning of bone marrow. We also have a proprietary process, as Tony mentioned earlier. We have spent a lot of time thinking about our manufacturing process, such that we can maintain the stemness of the cells that we extract from these patients. So those three things combined puts us in a great place from a process, analytics, from an efficacy, safety standpoint. The next question is, how do you translate that into manufacturing paradigm for larger number of patients, for global patients, et cetera, et cetera. And we are thinking through that. In fact, the approach we have taken is this hybrid approach of hiring people internally. We have hired a number of people who are experts in this space, people who came from developing products from at Juno, Novartis, Sarepta, and other places where they've done a lot of work on gene therapy, and we've hired those people. So these people internally develop the process for us, analytics for us. And then our idea would be to contract the routine type of manufacturing, GMP, testing type of activities to partners. Companies like Lonza, Aldevron help us in that regard. So that's what we are looking at. So again, it's, as Tony said, it's not a one-size-fit-all answer to this. Mm-hmm. And in fact, we have a second program, which is called hypophosphatasia program. This is a bone disorders. These babies who are born with that, and Tony knows this very well, working with Avaxis on SMA. These babies, if you don't treat them, don't grow into even infants. So in that regard, this second program we have is an exciting program, and we are looking at various ways to do manufacturing for that. But again, the long answer short, this is a process in evolution, and we're going to look at everything in our power. If we have to put up a small footprint of manufacturing, we will look at it. Mm-hmm. But majority of this is going to be in a hybrid format, get the internal resources aligned through the higher value activities, creating IP, et cetera, and try to get the routine type of activities outsourced, such that you can have the best of the both worlds, if you want to say it that way. So that's where we are.
Yeah. Palani, that's really interesting hearing that strategy around bringing in the talent. I really like that. So, as you know, and Matt, but PF, and we've managed over $4 billion in capital in the last 4 years, right, in manufacturing. So we're laying out the strategy and executing it. And one criteria when we're evaluating, whether it's internal or external, doesn't matter. We lay out the plan, ignite external, build internal, do both. Evaluating the team's talent is a great, indicator, if you will. So it's, okay, do I have the capability? Should I build internally? Or do I have to bring in all that talent on its own? Mm-hmm. So I really like the fact that your organization is, looking at ways to bring in that talent. So should you be provided that capability or that technology, that platform, you can actually use that, tool, if you will, to manufacture what you need. So really,
Tony, I'll take that opportunity to throw a little plug out. You not too long ago, you and I and your CEO Anshul Magal sat down and did a talent crunch roundtable. The listeners who are listening to this on the Bioprocess online website right now, you're only a couple clicks away from that very informative program as well that talks about overcoming your talent challenges. So check that out. Yeah, all right, plug aside. Palani, I wanted to ask you about something you mentioned just a few minutes ago in your response to the insource outsource question, and that's the regulatory landscape. So, depending on your perspective, that's a limiting or enabling pressure on gene therapy development. I think the word that most often is referenced or used in conversations with leaders of gene therapy companies around the regulatory scene right now is dialogue. Collaborative. What's your perspective? Where are you now on the regulatory path, and what does that regulatory path look like? From my understanding, it's sort of unfolding before our very eyes.
Yeah, a great question. I'm going to be a little bit of a contrarian to answer that question. If you ask a general answer to that question, people are going to say, "Hey, regulatory landscape is changing tremendously." And so you got to react to it and so on and so forth. In fact, my position in this is the regulatory landscape is not changing at all. We are interested in safe drugs for a long period of time. Doesn't matter, small molecule, biologics, antibody drug conjugates, microbiome, doesn't really matter. We want any external agent that comes in to intervene in a disease setting, we want it to be safe, We want it to be efficacious, and we want people having diseases derive benefit from this external agent. That position has not changed at all. And in fact, if you go back to talk to the regulators these days with your gene therapy, which I've been part of many of these regulatory meetings, the primary concern there is your agent safe? Is it efficacious? Can you measure the dose? Can you show that you can control the process? All of those things are the same. So that said, my point in all of this is that, end of the day, we have to have that dialogue with the regulators to be able to say, what we say in terms of the quality of the product, identity of the product, the impurity levels in this product. And I would even venture out to say we have to be even more careful because some of these impurities, we don't have the technologies to measure them yet. And these are genetic material. These are small quantities. And we got to be able to find those tools in our toolbox to be able to measure these things. And then I think we're going to be in a great position. From a regulatory perspective, I think that's going to be the key theme. And we know what genetic changes can do for pharmacologically in our system. So we just have to be careful about that. From that perspective, I maintain that, in fact, the regulatory landscape is evolving. I wouldn't call it a change, but we just have to take care of our business, end of the day.
Yeah.
Tony,
well said, Palani. I think you're right. Fundamentally, we're there. Interaction with the agency is critical, Matt. Like you had, collaboration and, upfront discussion, whether it's your CMC strategy, your regulatory strategy, your clinical studies and clinical strategy, right? But, and look, the agency wants us to be successful, right? They want to collaborate, especially with the meaningfulness behind this. If you zoom out and think about the industry, how did we get to where we are today? And Palani and I have had many chats around this, but we started out with therapeutic revolution, right? We started out with ultra rare orphan disease, incurable diseases, devastating, life-ending diseases. No other option, no other, potential care, solutions out there. We've really done a great job. We're starting to prove that out, whether it's, through the muscular dystrophies, the different types of derivations of cancer, or, different types of blood disorders. Now we're getting into these chronic treatments, right? And we're trying to find ways to cure these chronic treatments. And we're evolving, right? Society's getting comfortable with this, right? The regulators are getting comfortable with this. And so the patients have hope. Now we're moving into the chronic treatment. And eventually we're going to get into more pain management type of solutions, all using, cutting edge technology. So I think the progression is happening. And with that increases the patient population, right? Ultra rare, orphan disease, hundreds of patients, thousands, hundreds of thousands and millions. It'll come. And I think we've shown that in the regulatory pathway also, they're getting more comfortable with how we approve this and get that long-term data, as Palani highlighted.
Yeah, if I may add one more thing to that discussion, earlier paradigm is, you go test the drug in multiple countries, multiple populations, hundreds of patients, often thousands of patients. That's not the case anymore. We test these drugs in, several tens of patients and, sometimes hundreds of patients. So really there's an obligation on our part to be able to say that the success that you see in this early clinical population translate into commercial population. And again, the idea is, how do you manufacture in scale for these patients in that scale that we talked about, several hundreds of patients, perhaps in a commercial setting. Regulatory conversation in that regard, how do you validate your process? How do you set up the supply chain? Things like that becomes critical, going forward.
And diagnostics, right? Proper analytics.
Exactly, right.
That's a key one that needs to be, progressed alongside. So,
yeah. Tony, what manufacturing technologies are you seeing sticking right now? What I kind of want to shift that conversation to the tech that's being deployed and that's I guess being adopted and accepted as we kind of work through the manufacturing challenges that we're facing.
Yeah, thanks, man. They've continued to progress on their own right, but prefabricated modular clean rooms, right? I think that'll continue to stay and be of value, or just kind of, standard stick-built designs that can be modularly scaled out. Single-use technology or disposable technology, I think that's really valuable to avoid, the upfront effort related to steam in place, clean in place, needs. So those are there. Also, the other manufacturing benefits of like speed, fewer resources, smaller footprint, upfront costs related to that. I think that's going to continue to stick, and it will continue to progress. I mean, look, we worked with some of the first ever adhesion and suspension technologies in the space. We worked with some of the first ever modular clean rooms in the space. We actually built out the largest footprint for one of the vendors, all modular, clean rooms. And I will say 5 years ago to now, totally different in a good way, right?
Yeah.
They've really done a great job of hearing the voice of the customer. Palani and I have sat down with them and said, hey, these are really good things. These are areas of improvement. And, the technology, the equipment's technology side, like there's a science technology, there's a manufacturing equipment technology, has continued to improve. We've streamlined some of that. And now that those offerings, although they still are growing, and it's fun to hear, Matt, like folks, have their feedback on different technology. And I look and say, you should have seen it 5 years ago. A lot better now, right? And it'll continue to improve. Right. But I think those are there to stay.
The Business of Biotech is brought to you in partnership with Cytiva. Together, we're committed to helping the leaders of new and emerging biopharma companies navigate the financial, organizational, human resources, and regulatory waters you'll encounter on your way from discovery to the clinic and beyond. Check out a host of useful resources for biotech leaders at Cytiva's Emerging Biotech Accelerator at cytivalifesciences.com/emerging biotech. That's CYTIVA lifesciences.com/emerging biotech. What's been the scene over at Aruvant, Palani? What manufacturing technologies, I guess, since you've been there, or shortly before, that you've adopted as you've ironed out manufacturing issues? What's sort of been instrumental or game changing?
Oh yes, absolutely. That's a great question. I think we are in a very unique position because we are using, both a lentivirus based modification of the autologous cells for the sickle cell disease. And on the other side of the paradigm, we are also using adeno-associated virus as a way to inject these virus particles carrying the transgene in vivo. So we are testing both approaches in our programs. And that said, I think one of the areas that we are very carefully evaluating is this idea of how to grow these cells to make, a lot of virus particles, just going back to keeping it qualitative. Each of these cells, we call them the embryonic kidney cells. And these cells take up the transgene and take up the other genes and make the virus particles. And the virus particles contain the transgene that produces the protein that you're interested in, end of the day. And so we are looking at ways of enhancing to produce more virus particles carrying the right transgene. What does that, how does that matter? You have more virus particle carrying the transgene, you have to use less of them as a dose. More virus particles as a dose creates problem because it's foreign. Your liver has to work overtime to clear them. So lesser dose is better. So that's one area of interest for us. Can you produce more? Can you decrease the cost of goods for the patient? Remember, each of these doses is going to come at a premium price. And we've seen that throughout the industry. And so our goal would be can we decrease the cost of goods? Can you make these things, highly pure? And can you reduce the dose such that you don't have to create safety issues for these patients? Those are some of the areas we are interested in our shop to be able to identify solutions such that you can carry forward with these things, keeping or increasing the efficacy, but decreasing the safety, risk for the patients.
Yeah. Tony, what
qualitative answer, but I think we can get into details if you like, but that's just a qualitative way of looking at it, Matt.
Sure. Yeah. Yeah, I'm curious where you both see. I was gonna ask Tony as he sort of reflects on your response, Palani, based on his experience working with several gene therapy companies across the board. What your thoughts are on where we're going from here? What needs to get better? More specifically, where are we identified opportunities or room for improvement that's going to be necessary to move the ball forward? And Dr. Palaniappan, you're welcome to chime in on that as well. What's the next step in terms of making things better?
We got a lot to do, Matt. But we're moving towards it. I guess where to begin. I think Palani hit on it from a yield perspective, right? From a science maturity and trying to have better yields. Those better yields would result in higher, less batches, less footprint, better throughput. I think that'd be a great win. Looking at our technology and we talk about all these different single use that I brought up earlier and modular. Well, now the disposables are becoming long lead, and now we have these long leads that we talked about 5 years ago to now, and then we have a category called new long leads. And basically, we've got single-use disposables or filters and vials that are almost becoming equal to or greater than the lead time of a piece of equipment. And so we have to find ways to understand how this supply chain is continuing to ripple effect down and create more lead times. And that all comes with better forecasting. I think if we give the suppliers and the I call them partners, I think everybody's a partner. Everything we do takes a team and a huge industry movement. We give them a forecast of what we really need. I think that they'll have more confidence in scaling up and scaling out, if you will, so that they can service that. So we definitely need to do that better. Talent-wise is a bottleneck for sure. We're doing a good job investing in universities and trying to find ways. Training talent is a very unique creature, if you ask my perspective. Like, you could teach someone, they may remember, right? But if you involve them, they'll truly learn it, right? And so from my perspective, I think having apparatuses and having different solutions out there that allow university students or early phase training to physically touch systems and run equipment and be in the middle of it all, that could really help the talent bottleneck. And bottom line is we have a boom in the space and we have limited talent, so we've got to find ways to increase that and level out that discrepancy there. Yeah. So just a few things, and of course the technology menu, more automation, maybe more closed systems. I think we're doing a good job, but we definitely need to standardize things more and continue to progress on that forefront. I think that would bring a lot of value to the space. And right now, these processes are very manual.
Right.
Run this, it's almost like a glorified lab. Palani knows this, Matt. And if we can find ways to automate it, close those systems up more, and I always say mitigate your integration points. So all this equipment that's on the floor, integrating into a supervisory control system. And I'm a controls engineer by trade in the beginning of my life, so I won't geek out too much, but finding ways to have everything be more standardized and less custom integration, which results in custom testing, which extends lead times. And I think that would really benefit the space.
Yeah, I like the phrase mitigate your interfaces or integration points. That's a fantastic way to say that, because there are so many steps in this manufacturing, relatively speaking, right? In a small molecule biologic setting versus this one. Mm-hmm. If you don't mind, I would like to drill down a little bit. I think that was a great summary from a macro perspective. From a drilling down from an innovation perspective, I see three areas sort of come to my mind. We have to be more specific with these drugs. What do I mean by that? Tissue tropism. You have a range of, we look at range of diseases. When I was in Sarepta, we were looking at Duchenne's muscular dystrophy, a range of other dystrophies, muscular dystrophies. And people in different laboratories are looking at CNS disorders like Huntington and Parkinson's and things like that. In Aruvant, we are looking at sickle cell disease, hypophosphatasia, diseases like that. They are all different diseases and they target different tissues. And if we can have doses and virus particles that can specifically target these tissues, you eliminate the risk of bystander effect. The virus particles are not going to interact with other tissues in the body, thereby eliminating the safety risks that come with that. And we have seen insertional mutagenesis and clonal dominance in some of these studies. And that's going to be the huge issue for this gene therapy field going forward. So number one, be more specific and targeted to tissues. And if you can find virus particles that can do it, that's fabulous. Number two, I would like to decrease the dose. Right now we are putting millions of cells or millions of virus particles as a dose, and these need to be cleared in some fashion. Kidneys, livers work overtime in these patients. We got to be able to do that and decrease the dose so that first, you can keep it safe for the patients and efficacious. And second, you can decrease the cost of goods. So the amount of money people pay for these doses becomes lesser and lesser. What does that do? It sort of takes the drug and gives access worldwide. Because remember, you can charge patients in one market one way, but you can't charge patients the same way in other markets. You got to be able to make the drug work for everybody in the world. So I think that's going to be the key themes in my mind. Tissue tropism and keep it specific and targeted, decrease the dose and decrease the cost of goods and give access whoever needs that drug worldwide.
Yeah. Tony had mentioned just a few minutes ago that this is, many of these processes to meet those ends that you just mentioned, Palani, are still manual, and that there's room for improvement around automation. So, and I almost hesitate to ask this question because I know that the gap between achieving the things you just mentioned, specificity, tissue specificity, and decreasing dosage, a lot of that is a manual learning exercise. But is there an opportunity, or are companies right now leveraging advanced technologies like machine learning, artificial intelligence, to kind of address those things and help along the way to get closer to the answers?
Yeah, absolutely. I think that's where the industry is going. If you look at the number of companies that's come about in the Cambridge area in the last 2 years, a lot of them focus on machine learning. Companies that came out of MIT and other places focus on using machine learning, algorithms to be able to identify those types of capsids, different types of capsids that can target select tissues, and different types of capsids that can cross the blood-brain barrier, things like that. So there's a ton of effort going on. And, are we going to have success overnight in this space? Perhaps not. But as we continue the effort, I'm very hopeful. In the last, few years, we saw the tremendous amount of progress we made. In the next few years, we're going to see a tremendous amount of progress that will be made in the space of making it tissue tropic, making it low dose, making it impure, pure, and things of that nature is going to take a lot of focus as we go forward. Remember, there's about if you look at the rare disease space, there's probably 8,000 plus diseases out there, and we got to tackle all of them someday. We are probably focused on 10% of that 8,000 plus diseases today. We got the opportunity is unlimited here to be able to use machine learning, artificial intelligence to tackle some of those challenges.
Yeah, Palani, you actually reminded me of struck a memory for me. So, Matt, I don't know if I ever shared this with either one of you. So probably 3-4 years ago, I was invited to the Deerfield CEO conference. Basically, Deerfield has an annual event. Deerfield Management, it's a private equity fund focused on the healthcare space. I think they manage over 12 to 14 billion actively. But I was invited to attend that, and there was the keynote speaker. His name was Dr. Siddhartha Maharaji, I believe was his name, and he's a prize winner and author of The Emperor of All Maladies. Okay, and he gives this phenomenal presentation, like so enamored, and what he hit on, which was his opening, lines, if you will, where he showed his best friend at a wedding. This comes back to AI, so I promise I'm not going off the deep end. I'm staying focused. So
no, you know me, Tony. I'll go down any rabbit hole you want to dig.
yeah, exactly. I'm kept staying. So either way, he shows his best friend at a wedding. He attended the wedding, and he had what the doctor diagnosed was just a mole on his face. Thought nothing of it. 6 months later, cancer. 1-2 months later, passes away. And so Dr. Siddhartha was like, I'm all about this, focused in. How did we miss this? And they put it through AI. So they did this, software, and they trained the software to be able to identify cancer, and basically they were able to run his photo through with millions of other photos and identified it as cancer. And if they would have caught it then, something that wasn't known to the human eye, they would have been able to treat it, and that individual would still be with them today. And that struck a chord with me and drilled into my mind how exciting and important AI will be from a manufacturing, but also from a diagnostics perspective, to see who really needs help and when to catch that early. And
sure, yeah, and I think to the point I was making earlier around Palani's observances of things that Aruvant is working on, and that need to happen around dosage decreases and improving the efficacy of lower dosage, better tissue specificity. It occurs to me that there are great applications for artificial intelligence there, dependent on the data inputs, right? I mean, the data, to your point, Tony, there's got to be a whole big database to work with to be able to identify, potential risk in facial recognition or mole recognition. So, it's all data input dependent.
Right. You have to train that artificial intelligence, right? So it can identify. It's no different than, PF, we do manufacturing strategy and execution. Every time we execute, we re-input the data to have better strategy. It's this constant, engineering P&ID loop, right? But constant loop of enhancement. Same thing with AI, right? We got to pull all this data of non-cancerous to cancerous, images, and see, the system can be trained and captured early. So the data's got to be there for sure.
Exactly. The disease that we are dealing with these days are so heterogeneous, right? And I think there may be 1 or 2 or 3 or 5 patients worldwide on some of these diseases, and your ability to detect them and treat them appropriately in a personalized manner is going to be a key one. I'm not saying that's going to be the standard paradigm, but there's going to be situations like this in some of these rare diseases that we'll have to deal with. So artificial intelligence is going to play a major role in this setting going forward. That's just my hope and expectation as well.
Yeah. So we're running a bit short on time here, but I want to crank out a couple more responses from you two, if you'll indulge me as we wrap things up. Tony, I want to get your perspective on the movement towards non-viral delivery methods. I know that's been a hot topic lately. How do you see them non-viral? How do you see that playing a role as we move forward?
Yeah, I mean, it's an exciting, situation that we're in. And look, I mean, I think, the viral vector solutions are here to stay, especially for the, short to midterm. We have a proven solution, and we can penetrate different parts of the human body that we couldn't previously to carry, different genes and get them access to the, past the blood-brain barrier into a systemic solution or whatnot. But the non-viral solutions, I think that they will continue to improve. I think they're going to play a critical role in upcoming years. The biggest thing for me that's interesting and kind of zooming out is right now when we have a viral vector solution, there's also there's companion diagnostics to verify if people are the right fit for the the treatment of the drug. There's also post-treatment where you build up an immunity. Right. And I think with non-viral, solutions out there, delivery methods, there's an opportunity to maybe have one or, we only think about one treatment nowadays, but what if you needed multiple treatments to save a life and there are different diseases? If your body has built up immunity to it, you may not be a candidate to receive another dose of something else that could help. So that's what's very exciting about the non-viral side. I mean, the non-viral side compared to viral vector, right now there's low cytotoxicity, immunogenicity, attracting more researchers to explore like how we can promise delivery systems and move the gene therapy forward. The non-viral also does not carry ideal characteristics and has faced critical challenges such as the gene transfer efficiencies, right, and the specificity around that and the safety. So I think that it's got some areas to improve, but non-viral vectors have a fast, paced research topic in gene delivery, and I think that they'll continue to improve. The viral vector right now, we found that we've got a proven solution. We've got great data. It's gone through the regulatory pathway, put into humans, and it's continuing to show good success. And I think that'll be great. But there's also, drawbacks to that, right? So, understanding the long-term effects. We've long used viral vectors previously, right? And we've had catastrophic experiences with it. And, that's where we have our adeno-associated virus now instead of just the adenovirus. And, I think that there's going to continue to be more enhancements on both forefronts, and they'll both play a good role. Palani, any thoughts from your end?
Yeah, absolutely. We've talked about it quite a bit in terms of, my background with non-viral delivery goes back to my Gilead days very early on. I worked for a small company called Nexstar Pharmaceuticals, which was eventually acquired by Gilead. And at the time, we were thinking the lipid bubbles that we call, LNP these days, was used to contain drugs like amphotericin B and daunorubicin and doxorubicin to make them safer. Because these drugs themselves, by themselves, are very toxic drugs. So we wanted to shield them in a bubble so you can get them to the right tissues and so on and so forth. The industry has come a long way, and we've called them AV0 or LBV0 in some ways. That sort of says what we mean by that, right? And the key goal here is to get the transgene to the right tissue. And there are, particles that do that these days. And we saw a great data from Intellia and Regeneron over the last weekend of injecting these particles in vivo and creating the Cas9 needed to do the gene editing in vivo. I think things like that are happening. But still, I think as Tony noted, the virus particles are the natural way to get this to the tissues, and viruses know how to get to the tissues. We all know that, right?
Right.
And so I think this is here to stay. But we will make tremendous progress given the success that we have had with the lipid particles over the years. And I have tremendous expectation from that field as well. Going forward to make it more specific, more targeted, more tissue tropic, and so on. So same challenges, right? Doesn't matter virus particles, doesn't matter non-viral. The challenges are same. The key question is going to be, what sort of progress can we make to meet those challenges that are broad.
Yeah. Big challenges, but big opportunities.
Yeah, exactly.
Palani, the Intellia story is really exciting. I was speaking with some of our colleagues at Blackstone Life Sciences about it. And talk about a massive movement around CRISPR and gene editing in the space and how just promising and exciting that can look, and with the great data that's coming out and modifying in human type, genes and things like that, that could really change the, landscape of how we treat people and manufacture, if you will. So it's super exciting, super promising for sure.
Yeah.
No question about it.
Thanks for bringing that up.
So to wrap things up, like I said, unfortunately we have to. I feel like we could talk for another hour. We might have to do a part 2 if it's okay with you guys. But I want to wrap things up by inviting you, Palani, to share with me. Hey, you've covered a lot of ground today, but what if you had to sort of crystallize what you're most excited about and maybe what you're most concerned about moving forward? What would those be?
Yeah, so I think just to address your second question first, I think the thing I'm excited about is the opportunity to be able to treat any disease, right? Using gene therapy paradigm or cell therapy paradigm or in vivo gene therapy, cell therapy paradigm. That's a great opportunity for all of us. I think the diseases are here to stay, and they're going to increase in number, and we got to come up with a way to deal with that. That said, one thing that I worry about all the time is we seem to be in this idea that we are just one event away from everything breaking down, falling apart. I think we need to get past that idea. That's what keeps me at night. And on top of that, we talked about a couple of other things in the space. One, I worry about access. When we develop a drug, this is going to be accessible worldwide. Particularly for me, knowing sickle cell disease and the epidemiology there. It's an African disease. It's a Southeast Asian disease. It's a Mediterranean disease. Can we get this drug to worldwide? That's another thing that keeps me at night. So which has a lot of components in it, cost of goods and dose and all of that. And last but not least, I worry about the talent pool. Tony touched on this earlier. We can train people, but we need a workforce that's well-versed. In the 90s and 80s, I don't know if you remember this, we built biologics programs, interdisciplinary programs in many of these universities and institutions that brought in workforce, knowing what to do with biologics development. I think we have to take that approach now. The talent pool is small, and everyone is going after that same talent pool. And that is a differentiating factor in anything we do. Technologies can come and go, but the talent pool, the people are going to be here to, stay. And that's a differentiating factor for many of us. I feel like that's going to, we are sort of getting thinner every day on that.
Yeah, Palani, like I always, in our organization at PF, I'm always, imploring, tell me and I forget, teach me and I may remember, involve me and I learn. And I keep, you've heard me say it many times, and just having engineers and team members and project leads involved in different strategy discussions that usually they wouldn't be involved in, and just having them be, courtside seats, if you will, to that, to just mitigate the talent bottleneck. And the same thing needs to apply across the industry. We need to involve, our talent so that they can learn, and there's many ways to involve them. And then just, my most exciting thing, I think you've heard me, share this with both of you, is, again, it's a privilege to be a part of this industry. If you haven't seen it yet, go watch The Ataxian. It's a great movie. It's about Friedreich's ataxia, raises awareness of one of the many muscular dystrophies that we're all trying to cure. But just being a part of a therapeutic revolution where we can all work together as a team, lock arms, and cure incurable diseases and save and create lives, there's nothing more rewarding than that. And it doesn't matter, however we get there and however we do it, it's a privilege, it's an honor to be a part of that. And we can do a lot more. There's a ton of science that we can cure, that can be used to cure these devastating diseases. And it's a basic human right for humans to, I believe, have a good quality of life or be able to have access to these medicines. And that applies across the world. So we got to do a better job of getting access across the world, making things more cost effective for those, different, populations that need these treatments and these cures. And manufacturing is a key input, the science, the clinical trials. There's a lot of things that we can do collectively to really, make this a solution for everybody.
Yeah. Well, it's incredibly important work that you guys are doing. And I if it's appropriate for me to do so, I want to thank you for losing sleep, Palani. And I want to thank you for letting these things bother you. It's brilliant minds like yours that are solving these problems. And the fact that you're concerned about them is, it's wonderful. And we appreciate the work that you're doing.
Yeah, it's great to be sitting down with you, Matt and Tony, and the rest of the group.
Yeah,
thank you very much.
Our pleasure. So that's Aruvant CTO, Dr. Palani. Say it for me.
Palaniappan.
Palaniappan. All right. See, I got it right after I repeat right after you. I got it right. Dr.
That's a great collaboration there. Thank you.
Yeah, that's collaboration at its finest. And Project Farma EVP Tony Khoury. I'm Matt Piller, and this is the Business of Biotech. We're produced by Bioprocess Online in partnership with Cytiva. Go sign up for my newsletter at bioprocessonline.com. Then go to scientivalifesciences.com/emerging biotech to explore a trove of custom curated content designed to help guide the emerging biopharma's journey. And if you like what you heard here today, hit that subscribe button. Give us 5 stars. And as always, thank you for listening.